This is the third and last lecture on DNA metabolism. In this lecture, we discuss DNA recombination. Segments of DNA can rearrange their location. This can occur within and between chromosomes. Such recombination is involved in many biological processes that include repair of DNA, segregation of chromosomes during meiosis, and enhancement of genetic diversity. In sexually reproducing organisms, recombination and mutations are two driving forces of evolution. Recombination of co-infecting viral genomes may enhance virulence and provide resistance to antivirals. There are three classes of recombination: homologous or general recombination, site-specific recombination, and DNA transposition. In homologous or general recombination, there is exchange between two DNA molecules that share an extended region of similar sequence. In site-specific recombination, exchange occurs only at a particular sequence. In DNA transposition, short DNAs can move from one chromosome to another. These are jumping genes. The three known functions of homologous recombination are assistance in DNA repair, linking of sister chromosomes to properly segregate them between self and daughter cells, and a source of DNA exchange and genetic diversity. Here we're looking at homologous recombination in bacteria. You saw this example in the last lecture. Prior to step 1, when a replication fork encounters a break in one of the template strands, one arm of the fork is lost and the replication fork collapses. In step 1, the 5' end of the leading strand at the break is degraded to create a single-stranded 3' extension of the daughter strand. In step 2, there is a strand invasion from the daughter of the lagging strand. Note that this invasion repairs the DNA. Also note that both daughter strands are complementing one another. In step 3, the branch migration begins and continues in the opposite direction to which the replication fork was moving. This multiplex DNA structure is called a holiday junction or a holiday intermediate. In step 4, cleavage of the holiday intermediate but specialized nucleases, followed by ligation, restores a viable replication fork. The nick has been corrected, the replosome is reloaded onto the structure, and replication continues. The RUV A, B, and C complex is responsible for digesting holiday junctions. We'll talk about this complex in later slides. The REC-B-C-D helicase/nuclease complex. This complex processes DNA ends. It binds to a broken end and then unwinds and hydrolyzes the DNA. REC-B moves along one strand in a 3' to 5' direction, and REC-D moves along the other strand in a 5' to 3' direction. The REC-C subunit binds to the chi sequence GCT-GG-TGG and alters the activity of the enzyme complex. The chi sequence is a short stretch of DNA in a bacterium genome where homologous recombination is more likely to occur than on average across the genome. This cutaway view of the REC-B-C-D enzyme shows where the double-stranded DNA and separated single-stranded DNA bind. The subunits are shown in different colors. A bulbous protein structure called a pin, part of the REC-C subunit, facilitates the separation of strands. Activities of REC-B-C-D enzyme at a DNA end. The REC-B and REC-D subunits are helicases. Helicases are molecular motors that propel the complex along the DNA, a process that requires ATP. This is shown in step 1. In step 2, REC-B is shown to degrade both strands as the complex travels. REC-B cleaves the 3' ending strand more often than the 5' ending strand. In step 3, when a chi sequence is encountered on the 3' ending strand, the REC-C subunit binds to it, halting the advances of the strand through the complex. The 5' ending strand continues to be degraded as the 3' ending strand is looped out. This eventually creates a 3' single-stranded extension. In step 4, REC-A recombinase protein is loaded onto the processed DNA by the REC-BCD enzyme. There are more than 1,000 Chi sequences in E. coli. REC-A recombinase becomes active via the assembly of thousands of subunits. The subunits assemble cooperatively on DNA in steps of nucleation and extension. Cooperatively means that when binding of one subunit to DNA facilitates binding of the other subunits to DNA. REC-A protein filaments. REC-A and other recombinases in this class function as filaments of nucleoprotein. Filament formation proceeds in discrete nucleation and extension steps. Nucleation is the addition of the first few REC-A subunits. Extension occurs by adding REC-A subunits so that the filament grows in the 5' to 3' direction. When disassembly occurs, subunits are subtracted from the trailing end. This is an image of single-stranded DNA in complex with REC-A. This thin strand at the left of the image is single-stranded DNA, and the thicker strand to the right is single-stranded DNA bound to many REC-A proteins. Here is a filament of REC-A proteins bound to single-stranded DNA. The core of the REC-A structure is structurally related to domains in helicases, meaning that two proteins may have evolved from a common ancestor that was responsible for peeling double-stranded DNA into single-stranded DNA. Cleavage of the Holliday junction. I introduced the Holliday junction when I was talking about the use of homologous recombination to correct for a nick in DNA. The Holliday junction is a branched nucleic acid structure that contains 4 double-stranded DNA arms. RUVC is a specialized nuclease that binds to the RUVA-B complex and cleaves the Holliday intermediate on opposing sides of the junction, shown as red arrows, so that 2 contiguous DNA arms remain in each product. RUVA binds where 4 arms of Holliday junction come together. Hexamers of RUVB bind opposite arms and use ATP to propel DNA outward. RUVC is a helicase that binds to the RUVA-B complex. It cleaves the Holliday junction in opposite sides to create double-stranded DNA. A DNA ligase seals the nick. Meiosis in animal germline cells. Here we are looking at a diploid cell with 2 different chromosomes. Chromosomes replicate and are held together at their centromeres. Each replicated double-stranded DNA molecule is called a chromatid, sister chromatid. In prophase 1, just before meiotic division, the 2 homologous sets of chromatids align to form tetrads. These are held together by covalent links at homologous junctions called chiasmata. Crossover occurs when within the chiasmata. We'll discuss this in the next slide. These transient associations between homologs ensure that the 2 tethered chromosomes segregate properly in the next step, when attached spindle fibers pull them toward opposite poles of the dividing cell in the first meiotic division. The products of this division are 2 daughter cells, each with 2 pairs of different sister chromatids. The pairs now line up along the equator of the cell in preparation for separation of the chromatids, now called chromosomes. The second meiotic division produces 4 haploid daughter cells that can serve as gametes. Each has 2 chromosomes, half the number of the diploid germ cell line. The chromosomes have resorted and recombined. As a recap, homologous recombination occurs frequently during meiosis. Recombination occurs in the first meiotic division. Sequences called chiasmata are where the strand breaks and rejoining occurs. This occurs more often at hot spots. This process not only aligns sister chromatids for proper segregation, but it allows for exchange of genetic information, thus increasing genetic diversity. In the second meiotic division, physical links created by cohesins align chromatids and help them segregate properly. Recombination during prophase 1 in meiosis. Model of double-strand break repair for homologous genetic recombination. The two homologous chromosomes, as shown in red and in blue, involved in this recombination event have identical or very nearly identical sequences. Each of the two genes shown has different alleles on the two chromosomes. Chromosomes 1 and 2 align. A double-strand break occurs in chromosome 1. An exonuclease degrades the double-strand break ends to make a bigger gap. The 3' ending strands of the gap invade chromosome 2, where they pair with a complementary region on chromosome 2. This displaces a strand in chromosome 2, which ends up binding to chromosome 1. The displaced region of chromosome 2, now paired with chromosome 1, serves as a template to replace the missing DNA. In step 4, further DNA replication replaces the DNA missing from the site of the original double-strand break. This generates two holiday junctions. Nucleases called holiday intermediate revolvases cleave the holiday intermediates and generate one of two recombination products, product set 1 or product set 2. In product set 2, the DNA on either side of the region undergoing repair is recombined. Non-homologous end joining. This mechanism is used to repair double-strand breaks. The method is not ideal, but it's better than cell death. Unfortunately, there's no conservation of DNA sequence when the process completes. The Ku70-Ku80 complex is the first to bind the DNA ends. This is followed by a complex including the kinase DNA PKCS and the nucleus Artemis. DNA PKCS autophosphorylates, meaning that it phosphorylates itself and phosphorylates Artemis. Artemis removes the 5' or 3' single-stranded nucleotides or hairpins that have formed. A helicase separates the ends of the DNA, and X-ray repair cross-complementing protein 4, or XRCC4, or the alternative XLF, and DNA ligase 4 are recruited, either of two DNA polymerase, Pol-mu or Pol-lambda, subsequently extend the annealed DNA strands as needed before ligation occurs. Site-specific recombination. Site-specific recombination describes a variety of specialized recombination processes that involve reciprocal exchange between defined DNA sites. It is limited to specific sequences and uses recombinase enzymes with either tyrosine or serine as their nucleophiles. The reaction shown here is for a common class of site-specific recombinases called integrase class recombinases. The reaction is carried out within a tetramer of identical subunits, a homotetramer. Closely follow the color of the strands. Recombinase subunits bind to a specific sequence, called the recombination site. In step 1, diagonal subunits of the tetramer nick one strand of each double-stranded DNA, with the hydroxyl groups on the side chains of their tyrosine amino acids. The tyrosine is covalently attached to the 5' phosphate group at one end of the cleaved DNA, and the 3' OH of the cut DNA is released. In step 2, the recombinase ligates the free 3' OH of the opposite DNA strand with the DNA that is covalently attached to itself. In step 3, there is an isomerization in the holiday intermediate. In step 4, the other recombinase subunits cleave the two DNA strands that were not cleaved in step 1 through an identical mechanism used in step 1. In step 5, there is a rejoining of strands with chemistry that is identical to step 2, but with different partners. The final product is the site-specific recombination. Here is a surface representation of a Cre recombinase bound to a holiday junction. Effects of site-specific recombination. The outcome of site-specific recombination depends on the location and orientation of the recombination sites in a double-stranded DNA molecule. These sites are colored in red and green arrows. Note that the arrowheads show orientation that refers to the order of nucleotides in the recombination site, not the 5' to 3' direction of DNA. Figure A shows recombination sites with opposite orientation in the same DNA molecule. The result is an inversion. Figure B shows recombination sites with the same orientation, either on one DNA molecule, which produces a deletion, or on two DNA molecules, which produces an insertion. Resolution of Holiday junction can create dimeric DNAs. A special site-specific recombination system converts a dimeric genome into two monomer genomes. Remember that following the formation of a replication bubble, there will be two replication forks heading in opposite directions. In the event that a fork is undergoing recombinational repair, something we discussed earlier, a contiguous dimeric genome can be generated if cuts are made at the points indicated by red arrows, shown at the very top figure. In this scenario, a specialized site-specific recombinase called ZIR-CD in E. coli converts the dimer to monomers and allows the chromosomes to segregate for cell division to proceed. Transposable genetic elements. These are certain DNA segments that can change their relative position in chromosomes. These segments carry genes for transposases, but can contain extra genes for further functions. Example, antibiotic resistance. In bacteria, short, around 5 to 10 base pair sequences act as binding sites for transposases. Here we are looking at a transposon insertion into a target DNA. Should the transposase encoded by the transposon segment cuts the target DNA at specific sites. The transposon is inserted at the site of the cut, and the sequence flanking the transposon are duplicated. In this chapter, we learned that DNA replication is catalyzed by DNA polymerases. They use one of the strands as a template while adding new nucleotides to the 3' end of the newly synthesized chain. We learned several mechanisms exist to correct mismatches and other changes in DNA. We also learned that during DNA repair, the information encoded in the parent strand can be used to make corrections in the daughter strand. We learned that homologous recombination involves swapping of regions of DNA with similar sequences.